됐습니다. 네. 저부터 실시간 모니터링 하고 있겠습니다. 네. 딴다다단 다다단 다다단 단단 나다단 다다단. 박사님 들리시나요?
아, 네, 잘 들립니다.
목소리가 이렇게 힘이 없으십니까? 예?
아직 유튜브는 연결이 안 된 거죠?
유튜브 모르겠어요.
네.
한번 볼게요. 아, 유튜브 아주 잘 아이고 이거 왜 이래. 자, 잘 나오고 있습니다. 아주.
일찍 나와서
아, 네,
좀
좋습니다. 좋습니다.
어색하더라고요.
지금 동시 시청자 수 무려 5명이나 되고요, 벌써.
네.
네. 그 세 분이서 하시는 거죠, 오늘.
네, 맞습니다. 맞습니다.
네. 잠시만요.
저 이따가 들어오실
연결돼
겁니다.
있어요? 오케이. 맞습니다. 알파폴드 3 공개된 거 보셨습니까? API 열렸어요. 기터브에서 지금 마이크 안 들리시는데? 네.
아, 네.
네,
저 기침 잠깐
기터브에 공개가
나가지고,
돼가지고 알파폴드 3가 지금 이제 릴리즈 된 게 13시간 이렇게 돼 있더라고요. 그리고
네.
무슨 파일 하나 5시간 전에 올라와 있고. 그래가지고
음,
노벨상 기념으로 화끈하게 풀었다 생각했는데 뭘 다운로드를 뭘 받아야 되고 핵심적인 뭔가를 다운받으려면 거기다가 이메일을 보내고 뭐 이래야 되나 보더라고요. 보니까 그 넌 커머셜
네.
유즈고 해가지고 이거 어떡하지 하고 있네요. 예. 대격변의 시기입니다. 대격변의 시기 지금. 그 다른 분들
지금,
옆에
어,
김영순 박사님이신 것 같은데요?
네. 하이, 주로.
네?
하이, 다
마이클 상,
들어오셨어요?
안녕하세요.
아,
네.
안녕하세요.
안녕하세요.
Hi.
Hi, Dr. Jiang.
Oh, yeah, this
Right?
is
It's
Jerome.
good to see you. Is
Good to see
Hakjun
you
Kim
too.
at 진온바이오텍 in South Korea?
Great,
Yeah.
great. Thank you for your time.
Thank you. Thank you for your time too. And we're gonna start at 10 a.m. here. So it left 7 minutes. Is it okay for you?
네.
Yep. 황 박사님, 오늘 세 분이 연자신데,
네.
연자료는 한 명 것만 나간단 말이죠?
상관없는데?
뿜바이를 어떻게 하실 건지, 개인적인, 저기가 좀 있는데, 어떻게 하십니까? 이거 회식비로 쓰시나요?
네. 그렇게 했습니다.
네, 네, 알겠습니다.
네.
네. 발표 자료는 지난번에 보내주신 그 자료로 하시는 거죠?
아니요. 오늘 제가 화면을 띄울 건데요.
네,
거기서 아주 마이너한 수정이 있어서,
네,
그 제가 띄우고 하겠습니다.
알겠습니다.
네. 예, 혹시 공유 화면
네,
보이실까요?
잘 보입니다.
아, 네.
주로 이제
예.
들어오시는 분들은 대략 이제 상장사 대표님, 상장사의 상무님 분들도 계시고 비상장 벤처기업의 대표님들이나 연구 소장급 분들, 그리고 이제 박사님들이나 연구원 분들도 계시고요. 제가 이렇게 봤는데 이제 학사, 석사, 박사, 교수 이렇게 있으면 보통 한 박사급 정도 네,로 이제 판단됩니다. 대략 들으시는 분들은 네, 그 정도 되고 주로 이제 신약 개발 쪽, 당연히 주로 신약 개발 쪽 분들이 좀 많으십니다.
So, Zurong, the audience will be, I mean, mostly from academia at the level of PhDs and some of the students, as well as industry, I guess, directors and above,
어,
from very various companies.
first thing first, I must say hello.
Hi.
Hi, it's Hakjin Kim at 진온바이오텍. We're gonna start at 10 a.m. here, and then we left 2 minutes. And the audience are mostly PhD in the field of the pharmaceutical companies or pharmaceutical researches for the new drugs, and then mostly about 70% audience
Mhm.
are from the companies which is developing the new drugs, and then 30% are from the academy, maybe.
That's fantastic. Thank you so much.
No problem.
I think this is really a great opportunity that for us to give introduction on this topic. And if there's any interest question, we definitely would like to have some follow-up discussion. Thank you for organizing this format.
No problem. Thank you. It's my pleasure. And I have a question regarding BioAnalytx lab. There are 3 co-founders with 3 different locations, right?
Mhm.
So how is it making a synergy effect for the businesses for you
Yeah,
guys?
so we registered in state of Delaware in U.S. So we are a U.S. company. So three of us. So I'm as the CEO of the company. Ho-Sang is the Korean branch head president, and Jun is the chief technology officer. So, mostly we do have a lab facility in Korea. That's where Ho-Sang organized most of those activity, but we do have some consulting lab visit where a client in US, where June and I, we are both located in US. We will visit our client regularly.
So I understand that the wet lab, the research lab is in the South Korea,
wet
and
lab.
then two guys in the US are going to visit the customers and do consulting or something, making businesses.
Yes. Our current focus, at least at the first stage, is on the method development in the non-regular setting, but intended for regulatory environment. So our wet lab initially designed in Korea. But our second and third step would be, establish US wet lab for regulatory setting, because both June and I were, and as well as Hosang, we have supported those biopharmaceutical development, all different stage, all the way to the post-approval product at a commercial stage. So, we have collectively over 75 years experience. We could set up very easily. That's our near future plan.
Yeah.
Okay.
I mean, we used to all work together, and at least until last year, I guess Zurong also was residing in Maryland. So I'm in Maryland. So at least, I mean, we have been working since the time of when we were at Amgen in California, and then we transitioned to East Coast in AstraZeneca. And then we also worked for some, I guess, cell gene therapy company for a little bit. But during that time, I guess we also worked with Husang as one of the programs that we were leading at AstraZeneca for BioVentures. So that's how we got to know each other. But then, as I mentioned, Zurong and I, we were both in Maryland, and I think last year when Zurong actually moved back to California because of his spouse is also a scientist who found opportunity there in San Diego area. So that's how we kind of split it up in terms of location. But in how we conduct our business, I mean, we started our company as a mainly consulting company. So we do a lot of remote work, and then we do go do a site visits once the contract or once things are taking place. And then, so since we are doing consulting, most of the work, can be done remotely. But for last about a year or so, they have been asking for wet lab capability. This is where, we also asked Husang to see whether there's opportunity to set up a laboratory there, because obviously the expense is a big part. And setting up a laboratory in the US is much more expensive than in Korea. So at least for now, we're operating in this model. But once we have sufficient activity, and we do, it is in our plan to establish a lab in US site as well, because some of the companies are very sensitive about their IP and about their molecules. So some companies are not comfortable sending their materials out. So that's where we probably need to establish a lab here as well, in the US, as well as Korea.
Great. And the time is up, so we are going to start. Yep. First.
Right.
소개 먼저 하겠습니다. 안녕하십니까. 진온바이오텍 김학진입니다. 오늘 이 시간에는 우리 2회차 바이오어세인 드럭 디벨롭먼트 앤 프로덕트 라이프 사이클 매니지먼트에 대해서 바이오어널리틱스 랩의 세 분의 코파운더 분이 이제 돌아가면서 발표를 하시게 되었습니다. 그래서 먼저 황우상 박사님이 발표해 주시나요?
네,
네, 그러면은 바이오 아날리틱스 랩의 황우성 박사님이 먼저 발표를 해주시겠습니다. 먼저 간단하게 양력을 소개해드리자면은 황우성 박사님은 서울대 학교에서 연구교수, 그리고 차의과대학에서 연구교수를 하시고 삼성바이오로직스에서 근무하신 경험이 있으십니다. 자, 그러면은 박수로 맞이하도록 하겠습니다. 네, 부탁드립니다.
네, 안녕하세요. 방금 소개받은 황우상입니다. 저희가 아직 만들어진 지 얼마 안 된 회사다 보니까 아직은 팀워크가 좋지 않은 것 같아요. 벌써 이 제가 설명할 부분을 이미 좀 전에 이제 현준 선생님이 좀 소개해 주셨죠. 네, 그래서 저는 간단하게 이걸 언급하고 그다음에 발표를 진행하도록 하겠습니다. 좀 전에 말씀드린 것처럼 저희는 작년에 이 델러웨어에 만들었고 오피스는 지금 메릴랜드에 있는 걸 사용하고 있습니다. 그리고 현재는 일단 미국에 있는 회사의 컨설팅을 먼저 하고 있고요. 그다음에 웹랩에 대한 니즈들이 이제 생기는 것 부분들을 대비해서 또 아시아 쪽에서 이제 바이오 분야들이 발전하고 우리나라 포함한 중국, 일본 그래서 이쪽 부분에 대한 니즈들이 좀 있을 것 같아서 저는 좀 한국 쪽에 웹랩을 만들고 그런 일들을 같이 좀 해보고 싶어서 한국에다 이제 좀 준비를 하고 있습니다. 어, 좀 전에 설명드린 것처럼 저희 이제 지금 시행을 하고 있는 그 주롱은 아스트라제네카, 엠젠에서 35년 이상의 바이오세이 분야, 그다음에 QA, 퀄리티 매니지먼트 뭐 이런 부분을 경력을 갖고 계시고요. 그다음에 어, 두 번째로 소개해드린 건축 김현준 관련된 스탯의 전문가입니다. 쇼바이 차우는 에스트레네카의 SVP고
이거 두 개 껐어서.
프랭크는 여러분 바이오스에 하시는 분들이 서버를 많이 써보셨을 그 프로메가 그 회사에서 지금 바이오 디비전 헤드를 하고 계시고요. 이전에는 GSK에서 근무를 하셨습니다. 마지막으로 로린이라는 분은 바이오세이 필드에 되게 이제 유명한 학교가 베파라는 학교가 있는데 뭐 1년에 미국과 유럽을 왔다 갔다 하면서 1년에 두 번씩 여는 학회인데요. 그 학회를 하고 계시고 또 인더스트리랑 되게 많은 그런 인맥을 갖고 계셔서 이 분야에 되게 큰 발입니다. 그래서 저희가 어드바이저리 보드로 모셨고 그다음에 이제 우리나라도 점점 바이오 분야에 많은 성과들을 내고 있잖아요. 그래서 외국 회사들도 한국 몰레큐를 관심을 갖고 있고 또 그래서 그런 부분들이 로렌 이분도 이제 알고 있고 관심이 있어서 아마 머지않은 시간에 그 베파 아시아 그 컨퍼런스를 한국에 좀 개최해 볼까 그 생각을 하고 준비를 하고 있습니다. 네. 오늘 발표의 아웃라인입니다. 먼저 저희는 바이오스의 드럭 디스커버리나 드럭 디벨롭먼트, 그 다음에 드럭의 라이프 사이클 매니지먼트에 있어서 바이오스의 롤, 그다음에 바이오스 디벨롭먼트에 대한 그 레귤레이터리 허가기관의 익스펙테이션, FDA, EMA 포함이요. 그다음 약물 개발하는 과정에서 이제 베이스 어프로치를 주로 하게 되잖아요. 그때 어떻게 해서 바이오스를 페이즈만 맞게 디벨롭하고 라이프 사이클 매니지먼트를 하는지 이런 부분들을 이제 그 주홍이 설명을 해주실 거고요. 그다음에 실제적으로 이런 설명을 들어도 어떻게 적용하는지를 케이스 스터디를 알면 더 이해가 빠르잖아요. 그래서 potency assay development 하는 케이스 스터디 저희가 근데 뭐 이전에 있던 회사의 데이터를 다 쓸 수는 없어서 일단 ISH Q14에 나와 있는 내용을 사용해서 설명해 드릴 거고요. 두 번째는 potency assay가 저희가 이제 베이스 어프로치 하다 보니까 어느 순간 이제 어세이를 변경하거나 이런 경우가 생기는데 그럴 때 어떻게 이 메서드를 리플레이스먼트를 하는지에 대한 케이스 스터디를 또 하나 발표를 해드릴 거고요. 요거는 다행히도 이전 회사에 있었을 때 자료를 학회에 발표한 자료가 있어서 그 부분을 이용해서 김현준 님이 발표를 해주시도록 하겠습니다. 마지막으로 서머리 있습니다. Dr. Chung, would you please give your presentations?
Thank you very much, Ho-san, for your introduction. I really want to say my appreciation and thankfulness for Dr. Jing All opportunity. So I start with the role of bioassay in drug discovery, development, and life cycle management support. So on the top left side, this is really in the research discovery phases. Typically, you employ those assays, support research, and try to understand mechanism of action for the drug candidate selection, as well as some preclinical study where you link your in vitro MOA to the in vivo biological activity study in the animal. And then you move to the next phase, translational science, where often those immunogenicity, pharmacokinetics, pharmacodynamics, biomarker, those kind of clinical assay need to be in places where bioassay, especially cell-based assay, are often used for NAB assay assessment. That's typically involved in kind of a tiered approach. I elaborate a little bit more next few slides. In the area of CMC, where bioassay is a necessity and must-have for product characterization, that's typically factored into the regular filing, comparability throughout the clinical and post-approval stage. Bioassay always a must-have. It's also used for reference standard qualification, structure function relationship study, and also some of those formulation development in process control study sometimes involve those kind of biological assay support. And then in the more regular area on the left bottom side, where typically you need to use qualify or validate biological assay for the product release, stability testing. At the pivotal stage, typically you require a validated assay, but during clinical stage, usually you require a certain level of assay qualification. And sometimes the assay you develop not really intended for your use, you have to really transfer to manufacturer facilities such as CDMO, others. You do, let them, the QC lab run those assays. And sometimes they run into issue. You as a developer need to involve to do those kind of deviation investigation, ensure find the root cause. So there's a lot of a factor into those kind of detailed activity. Next slide, please. Right. So a little bit of elaboration on the early phase drug discovery. Typically, those assays is more used for proof of principle. Here you could see the top panel show the stage of development from discovery to the lead optimization, selection of clinical and clinical candidate to the IND and clinical development. So during those kind of early phases, typically you do narrow down approach. Number of candidates could range from 100 at the discovery phase, that narrowed down to 2 to 3 to 1, typically went to the IND and clinical stage. So how do you do that? So definitely you have to perform a proof of principle work, and all these stages involve the application of a biological assay. Ensure you understand target the disease MOA, and link those biological activity to the in vivo efficacy. Meanwhile, you will also have other physicochemical assay involved to do a developability study, such as you select clone that do not have a lot of sequence hotspot, sequence liability, have a developability issues such as form aggregation, are not stable or cannot be formulated at a high concentration, or cause pain. You do a delivery as epidermal. So there's a lot of things involved in each step. And then, once you select those one more skillful for the future development, then, you start the GLP manufacture of those material for the GLP tox study, as well as produce the first GMP batch. Next slide, please. So during those early stage, bioassay is really important to be used in defining your target product profile, quality target product profile, sometimes making go, no-go decision to the clinical study. Why is that? So during the lead target optimization phases, usually this biological assay is used to support the target selection and validation. It's also being used for the lead optimization and further characterization of the target molecule. For instance, if you have a therapeutic monoclonal with Fc effector function, or you're trying to engineer out through glycan engineering to reduce Fc effector function, but you need those assays to measure that. You typically use a binding and also use a functional ADCC assay, and sometimes those assays fairly variable. That's where you need to really ensure your assay fulfill your needed purpose. Otherwise, you might draw undesirable conclusion that's costing you tons later on. Also, you have to assess the PK immunogenicity to have a de-risking strategy. In other words, you have the right assay in place to do that. So that's really point to the direction. It's not just a assay, but a right assay that needed to do those evaluation to serve those purpose. And there is plenty example when those wrong assay used where lead to the wrong conclusion that could end up a loss of millions of dollars of investment. Next slide, please. So this slide actually is publicly available. It's really, I want to use it as an example. This is a largest biotech company, Amgen, when their proposed approach for the immunogenicity assessment. So typically immunogenicity, that's more in the area of translational science, where you do typically at the discovery, lead optimization, preclinical phase, in silico, do those kind of a sequence analysis where a lot of CDMO they could assist you to run those sequence analysis. You could also do some HLA typing to make sure you understand the sequence molecule, the attributes based on your assessment. And once you start the first time in human after the IND and clinical study, then a lot of those lab immunogenicity work kick in. Typically, they use those tiered approach with kind of like ELISA method to do those kind of immuno ADA assessment, but sometimes when there is a neutralizing antibody that could impact your PK and as well as safety profile of the molecule, then a bioassay, a cell base need to be in place to ensure you have the right assessment. Next slide, please. So I want just to say that even during those drug selection stage, there's always challenges, and there are a way to mitigate those challenges. For example, a lot of time you see screening assay, they are based on ELISA method that measure a binding to a trimeric ligand. And you know those assay conditions can cause dissociation, lack of epitope expression. On top, those binding assays is not capable of detect those transformational change ability of those molecules, eventually lead to a functional change. So therefore, if you have those kind of in place, some of those functional changes it might not be suitable for such testing. The result of this consequence of this is you really risk to miss an excellent drug candidate. So there's a lot of industry lesson learned. So better understanding of underlying mechanism of the disease or mechanism of action of the molecule can help identify the appropriate drug candidate. But if your bioassay used to identify select the lead candidate are inadequate, then it could result in failure later on in the clinical development. So this is easily said than done. But the consequence is very serious. Next slide, please. So come down to the bioassay use in the regulatory area, especially for product release stability. What is the regulatory expectation? So in the ICH Q6B, in some of the FDA guidance document, all universities say that your potency assay used for product release stability should be both quantitative and a representative of the product mechanism of action. So that's kind of a bottom line requirement. So we know that for biologics, including different therapeutic antibody, ADC, T-cell engager, or cell gene therapy product, they all need potency assay for product release stability. In addition, in your IND filing, you have to conduct extensive biological characterization study. So there is different level of biological characterization. And then ICH-Q5E, where specifically provide guidance on comparability study throughout the clinical development, all the way to the post-approval. You have to follow those where biological assay is one of the key assay used there. And this assay is the only test for functionality of the product. Next slide, please. So knowing there's this kind of guidance, how do you develop a bioassay? So on the high level regular expectation, you need to read this European Pharmacopoeia section 5, where the specifics say how to design and analysis of bioassay. Then there is also your USP chapter. A USP chapter above 1,000 is for reference, whereas below 1,000 is legally bound. So chapter 111, everybody have to follow when you have the product approved in US. You have to really use those chapter 111. Whereas the design biological assay and validation biological assay 1032, 1033, this is where I give you a kind of a guidance how you design your assay, how you validate the biological assay. Next slide, please. So this slide, I want to reduce those high-level expectation to a lab scientist, what you do in order to achieve a good biological assay where you are defendable throughout the product development. So on the EMA, FDA, other health authorities, you know, bioassay is the single most important analytical method for the product quality. So you need to make sure you have a best practice or strategy in place when to qualify your assay, when to validate the assay, and what guidance you follow to qualify, validate those assays. So at the end, after your assay transfer to a QC lab, and that's usually a measure how robust your assay is. So you need to demonstrate your assay have a lower assay failure rate. Otherwise, you are going to run into an issue. Is this measure accurate? So in a practical sense, industrial lab, let's say those quality lab in AstraZeneca, Amgen, or other big company. So what a desirable bioassay look like? Firstly, the assay should have a lower variability in general. Assay have a less than or equal to 15% CV. It should be high throughput and robust so that you could run more assays so that cost of good take into consideration. QC analysis could be easy to perform. Otherwise, you have such a sophisticated, they could make a lot of mistake. Therefore, you got a higher assay failure rate. So ideally, you have a shorter time, like the shorter you have, the higher, the quicker you could release the product. The cost could be reduced too. There are also other factors you need to take into consideration, such as don't use primary cell because those cells not only expensive, they also tend to be more variable, not QC friendly. Avoid to use radioactivity because there's so many technology you have a better alternative. And make sure when you secure a vendor or a critical reagent, you have a you don't really part with one vendor, they might go out of business. Please depend on commercial case in case those commercial case going wrong, you have a better control. You have a credible instrument from a credible vendor that need to be Part 11 compliance. And then, you know, bioassay, there is a very important factor is data analysis because those are European Pharmacopoeia Section 5 require you need to do the parallel analysis. So this software need to be validated or at least validatable. And then those methods should align with analytical life cycle management, meaning that, you know, when you transfer to a 2030 lab after the approval, those assays should be robust enough. Next slide, please. So this is more of a snapshot of the stage development where those potency bioassay development kick in, and what do you do at each stage. So typically the start of bioassay development in the CMC area is at the preclinical phases. Assay need to be in place for GLP tox lot release as well first GMP batch. So typically this assay need to be qualified, not validated at this stage. And sometimes taking a certain time to do a cell-based assay, some companies usually use a binding assay to start with. But one need to make sure that your binding assay should reflect product and mechanism of action. Otherwise, in your IND filing, you are going to be for that. Throughout the clinical development phase, we know those potency assays should be used for the release of GMP batches, doing stability study, contribute to the comparability, as well as biological characterization testing. So the stage start with pivotal, which typically end of phase 2, where you should select the mechanism of action reflecting potency assay as your final assay going to be validated. And then, you should use those validated assay for phase 3, especially the PPQ material and for the BLA filing. Any assay used for comparability post-approval, that should use a validated assay. Next slide, please. So when you develop a potency, how you should select your potency release assay. So first consideration, again, I need to emphasize that you understand your product mechanism of action, and based on your those. One example is if you have a product that's an agonist type of product, like a growth factor, endogenous protein, then you can a binding assay. You have to select a cell-based assay. But for a product like an antagonist product, such as therapeutic monoclonal antibody bispecific. So depending on product mechanism of action, if a class 1, class 2 monoclonal antibody molecule, you typically select a cell-based assay. So class 1 monoclonal mean the monoclonal effector function mechanism of action, such as rituximab. Class 2 molecule, typically therapeutic monoclonal, binding to a cell surface receptor or cell surface molecule. In this case, a cell base is more appropriate. Class 3 molecule, you could select a binding assay. Class 3 means your target is a soluble ligand, not a membrane associated. So the mechanism of action purely neutralizes that ligand. The second consideration for potency assay selection is the assay format sensitive to changing in the molecule? Is your assay stability indicator. Even though FDA not absolutely require at this stage, because there's a lot of things you don't understand. But if you have certain studies show that you have one assay more stability indicating than others, then you better select that stability indicating assay. Otherwise, the future changes, you have to basically spend a lot of time, spend a lot of budget, and could potentially delay your development timeline. The second question is, does the therapeutic have historical precedent for the use of bias, such as like interferon? If there is such thing exists, then you probably could either select those assays that are suitable for those purposes, or you have some other function assay that's reduce variability, you could select those. So keep in mind, mechanism of action is important for this consideration. Next slide, please. There are plenty consideration for assay development, such as the sourcing of cell line. Because at the early stage, you would think, yeah, I could just buy a cell line. But make sure when you buy that, the vendor would give you the freedom of operation when you move your product to a later phase, especially moving to the commercialization. Otherwise, when you negotiate with vendor later, you have to probably pay a lot of high fee. When you engineer the cell for yourself, you also need to keep IP evaluation into consideration so that you don't see surprise there. This is also true when you create your own monster and Make sure you understand and characterize fully of your working cell bank, even though cell bank used for product release, not used for product manufacture. And lastly, the sourcing of critical reagent is important. Make sure for business continuity, you secure multiple vendor, understand expiry date, ideally understand the stability of your reagent, and secure long-term supply. Make sure you have a process how to qualify this critical reagent. Next slide, please. And then, come down to the assay qualification. There are some guidance there, but this guidance is really on the higher level, like ICH Q2A, Q2B provide guidance on assay qualification, but they don't have solid assay acceptance criteria. So what we see industry typically do is you establish your in-house guidance to set up a target criteria assay qualification. For instance, like Amgen, we have our own internal guidance. At least you get a clarity what to expect. Number of assay, your target precision, accuracy, what assay format, how you ensure data analysis, even like how to build your analyzer macro or assay template, where we actually we publish in PDA journal where AstraZeneca, Amgen, they use those that actually save very significantly. Next slide, please. So it is important you collect data during those clinical development phases. The reason for that is, when you transfer method to the QC lab, you really want to ensure the real world assay performed at end user lab, the data being collected. What data to be collected? You make sure you have your assay control in the system. You understand those assay control trending. Also collect curve parameter, paralysis criteria, cell log, EC50, things like those. These are all important parameters for the assay training. So the criticality of the data training, it will be extremely helpful for troubleshooting of the method across different labs, especially when you do remediation in a different lab, try to find the root cause, what caused those assay malfunction. Reconfirm the method capability during method qualification. Allow distinction between method and process capability, because you likely during the process development, this method support, you are going to see those method as well process variation. So you have to really ensure your method robust enough so that when there is a process variation, you could detect that. Of course, there's other important things such as paralysis criteria, SOP revision for the method validation. These are all important based on data training. Next slide, please. So after analyze of those data training, they come down to the SOP revision. So, during those clinical development phase, the SOP is likely need to be revised during the life cycle support. So, typically at the validation stage, you do a validation enabling qualification of those methods to ensure the parameter you change, optimize, reflect in the revised procedure. And then those revised procedure will be in place prior to your method validation. Next slide, please. Also, gap analysis is important when you move to a later phase. So, especially before the validation. So, you need to ask a few questions. Have all ICH requirements be evaluated, such as your accuracy, repeatability, intermediate precision, so on and so forth? Has sufficient robust testing been performed? Has stability indicating potential method been evaluated? Method process variability from a historical DSDP log being evaluated? And does the receiving lab have all equipment necessary to perform the method? So there's a lot of things you have to work with the receiving lab to conduct those activities prior to the method validation. Next slide, please. And then you come to a very important testing called robust testing. So robust testing for the bioassay divided two areas. So first one is the multifactorial DOE performed at various times, and that typically before a method validation, before your method fully qualified, for the clinical study. And then, at the later phase, prior to the validation, then you do a validation enabling qualification, robust testing to ensure that after tech transfer a method, your method will be performing in a robust manner in the receiving lab that could validate those methods. Next slide, please. Come down to tech transfer. So you all know analytical method transfer is challenging, particularly to come down to the bioassay. The ease of tech transfer is direct proportional to the upfront work made during method development. For instance, you have to really make sure your transferring lab, receiving lab, where they engage, you conduct training at each lab. They have a pilot run of those methods. Make sure you have a transfer protocol in place, define the transfer criteria. And then after transfer is done, you write in transfer report to conclude those activity. But of course, training is very important. You have to really ensure your SME train those receiving labs so that they would be able to perform the method just like a developer. So an item need to be in place from the transferring lab, make the job more easier, is have an effective SOP, have a full method qualification package, have availability of long-term training data, have a robust testing. So those could really ease the tech transfer activity. Next slide, please. So this is probably the last slide of my presentation today. Before I hand over to Hosang, this is for bioassay validation. As you know, this validation typically start at the pivotal when you start before phase 3 start. Validation should simply be a verification of method historical performance and robust DOE study. So really, when your method qualifies robust, then validation should be simple, easier. At this stage, you shouldn't see surprise from a well-developed and controlled assay. Sometimes you will need to generate additional data for analyst to analyst and lab to lab analysis. For validation, you need to perform based on ICH Q2AB guidance that cover a range of like specificity, linearity, range, accuracy, precision. So that my presentation today, and I will happy for hand over to Hosang. Thank you.
네. 이제 케이스 스터디로 예를 들어 설명을 해드리겠습니다. 그거는 ICH Q14이고 2023년도에 파이널 버전이 나왔습니다. 잠시만요. 네. 어, 여기서 일단 이제 어떻게 그러면 바이오스를 개발할 것인가 그거에 대한 이제 전략에 대해서 이제 설명을 해드릴 건데요. 첫 번째는 이제 어널리티컬 타겟 프로파일이라는 걸 만들어야 됩니다. 저희는 이제 오늘 예로 TNF 알파 이니비션 어세이를 Q14에 나와 있는 걸 설명해 드릴 건데요. 먼저 이 어세이가 과연 그 인텐디브 퍼포스가 뭐냐라는 거를 확실히 알아야 되겠죠. 안티 TNF 알파니까 요거를 뉴트럴라이징 하는 어세이인 거고요. 그 다음에 이게 link to Q&A, CQA. 그러니까 이게 biological activity랑 어떻게 링크가 되어 있는지 저희가 이 부분을 사전에 잘 파악을 해야 되고, 실제적으로 이 어세이의 이제 타겟 억셉턴스 크라이테리아, 여기서는 지금 80, 125, 이거는 이제 어세이별로 개별적으로 적용하는 값이기 때문에 항상 이거를 동일하게 적용한다고 생각하시지 말고요. 80에서 125, 125를 적용해서 어세이를 만들겠다. 이런 것들을 일단 우리가 어떤 어세이를 개발할 것인가 목표에 접어두고, 그리고 그 세부적으로 accuracy, precision, specificity, reportable range 이런 기준들을 사전에 predefined criteria 를 가져가는 거예요. 그래서 내가 어세이를 개발할 때 accuracy에 대해서는 지금 보시는 것처럼 upper and lower의 90% confidence interval로 relative bias calculator 할 건데 각각의 그 potential 레벨에 대해서 요거는 20% 이하여야 된다. 이런 것들. 그다음에 precision은 저희가 샘플을 런해서 그 안에 95% CI로 했을 때 geometric CV가 20% 이하에 있어야 된다. specificity 여기서는 이제 버퍼나 아니면 이 약물의 그 어세이 반응이 스페시픽이 나오게 하는 부분이고요. 그걸 어떻게 해석해서 기준을 잡을 것인지를 기술해 주고요. 제일 중요한 거는 리포터블 레인지인 것 같습니다. 왜냐하면 이게 스페시피케이션과 관련도 있고 어세이 베리에이션과 관련도 있기 때문에, 그래서 실질적으로 보면 만약에 say specific range를 뭐 80, 120을 본다면 그것에 또 베리에이션이 있기 때문에 실제적으로 acceptance range는 이 베리에이션을 감안해서 64%부터 150%를 커버하는 say 이 안에서 quantitatively 잘 정량이 되는 어세이를 개발하는 걸 목표로 삼겠다. 뭐 이런 정도의 그 타겟 프로파일을 잡고 어세이 개발을 시작하는 것이 좋습니다. 자, 두 번째는 여기에 어세이를 이런 퀄리티로 개발하겠다고 결정했으면 이제 어떤 방법을 써야 할 것인가라는 고민을 하시게 될 건데요. 제가 설명을 좀 용이하기 위해서 한 예로 우리가 이제 그 파티클 그 측정하는 방법에 대해서 예를 한번 들어봤어요. 이게 가장 이해하기 접근하기 쉬울 것 같아서요. 우리가 알고 있는 파티클은 비저블과 서비저블 파티클이 있죠. 그래서 비저블 파티클은 뭐 의약품 릴리즈에서 쓰지만 비주얼 인스펙션, 눈에 보이는 거니까 100마이크로 이상의 그런 것들은 비주얼 인스펙션을 쓰는 방법이 쉽고, 그 다음에 인프라레드 마이크로스코피를 써도 되고요. FTIR 마이크로스코피도 어느 정도 커버가 됩니다. 근데 이 이하의 서비저블 베지클, 저희가 의약품 할 때 보통 서비저블 베지클을 10, 25 마이크로 기준을 가지고 스페시픽 많이 사용하죠. 그때는 사이즈가 10, 그 다음에 25 마이크로미터이기 때문에 우리가 릴리즈에 제일 많이 쓰는 방법은 라이트 옵스쿨레이션 많이 쓰죠. 그 다음에 그 항체 같은 경우에 이제 뭐 멀티머, 모노머 이런 거 확인할 때 많이 쓰는 방법이 사이즈 익스클루전, SEC, HPLC 메서드를 많이 사용하죠. 그리고 이거에 어서버널 메서드로 그 아날리티컬 울트라 센트리피케이션이나 그 다음에 페이스트 필드 플로우 프로큘레이션 AF4, 그다음에 LS 라이트 스카터링 이런 부분들을 우리가 어서거널로 해서 같이 해석을 하잖아요. 그 사이즈 크기에 따라서 이거 말씀드린 이유는 우리가 분석하고자 하는 그 목적에 맞게 맞는 방법들을 찾아서 어세이 개발하는 게 맞겠죠. 이거를 이제 바이어세이, 포텐시어세이 등을 적용하자고 하면 이렇게 TNF-알파 이니비션을 한다고 했었을 때 당연히 엘라이즈 방법들이에서 바인딩을 볼 수 있는 방법도 있을 거고, 그다음에 또 SPR 통해서 바인딩 볼 수 있는 방법도 있을 거고, 셀라이저 통해서 실제 셀 서피스의 반응을 이니비션 하는지를 볼 수도 있을 거고, 하나는 또 셀 베이스 어세이를 써서 이 어세이를 통해서 우리가 이니비션이 일어나는지도 볼 수 있을 거고, 다양한 방법들이 있을 수 있습니다. 여기서는 셀 베이스 어세이 예를 들 건데요. 실제적으로 또 셀 베이스를 우리가 한다고 했을 때는 실제적으로 TNF 알파에 의해서 발생하는 사이토톡시티나 에코피시스를 보면서 이니비션을 볼 건지, 아니면 또 리포터진을 써서 볼 건지, 이와 같이 다양한 방법들이 있을 수 있는데, 우리가 만들고자 하는 약의 MOA를 가장 잘 반영하는 어세이를 개발하는 것이 유리합니다. 그래서 이제 우리가 어, 셀 베이스 어세이를, 지금 이건 카메라메트릭 미서드로 개발하겠다라는 접근 방법에 있어서의 전략을 설명하는 건데요. 이건 이제 그 피시분 트리, 이시가 다이그램이죠. 이걸 했을 때 실제적으로 우리가 어세이 개발함에 있어서 어느 과정들이 필요한지를 다 이제 디파인 해놓는 겁니다. 그래서 셀 프레퍼레이션, 여기 안에서 실제적으로 미디어 프레퍼레이션, 미디어 뭐 쓸 건지, 일단 미디어 셀 서스펜션, 프리퍼레이션 어떻게 할 건지, 그때 뭐 템퍼러처나 또 이 어세이를 극명하게 보이기 위해서 뭐 액티노마이신을 어떻게 사용할 것인지, 그다음에 TNF 알파 레퍼런스는 이건 이니비션 어세이니까 요거에 지금 제일 중요한 건 TNF 알파 레퍼런스 물질의 퀄리티죠. 그래서 어떤 퀄리티의 어떤 물질을 어떻게 관리하면서 쓰겠다라는 전략들을 세우는 게 좋고요. 샘플 프레퍼레이션, 그다음에 여기 프로덕트 스페시피케이션 레퍼런스, 이 두 가지는 이제 또 레퍼런스나 측정을 된 샘플을 어떻게 관리할 거고, 어떻게 투여할 거고, 샘플당 외를 얼마큼 집어넣을 거고, 딜루션을 어떻게 가져갈 건지 이런 부분들을 집어넣어서 사전 예상을 할 수 있고요. 그다음에 calculation of reportable result. 자, 이거는 어세이를 했을 때의 베리에이션에 따라서 리플레이션을 몇 가지로 가져갈 것인지, 또 포뮬러 어떤 걸 쓸 건지, 커브 해석 어떻게 할 것인지, 또 패러리즘 어떻게 할 건지, 렐러티브 밸류는 어떻게 구할 것인지 등등 것들을 이제 고려해야 됩니다. 전에. 실제적으로 이제 어세이 익스큐션에 있어서는 셀을 얼마만큼 사용할 것인지, 파자티브 컨트롤 웰은 어디를 어떻게 써서 이거를 시스템 스테빌리티 확인할 때 어떻게 적용할 것인지, 또 TNF 알파 언제 얼마큼 양을 집어넣고, 셀 인큐베이션 하는 어세이 동안 인큐베이션, CO2 레벨, 또 인큐베이션 템퍼러처, 뭐 이런 부분들을 사전에 이제 고려를 해서 인정을 해야 되겠죠. 그 다음에 또 도스 레스판스 커브 컨스트럭션, 여기 관련해서는 지금 어떤 이제 그 리전트를 써서 우리가 할 것인지, 그때 또 인큐베이션 어떻게 할 것인지, 왜냐면 이런 것들이 다 그 도스 레스판스 커브에 영향을 준 거기 때문에 그것들을 우리가 어떻게 그 키네틱을 만들어서 커브를 그릴 것인지를, 이런 부분들을 고려를 해서 어세이 디벨롭을 디자인하게 됩니다. 실제적으로 이런 디자인을 하게 되면 우리가 이제 각각의 액티비티에 대한 이제 우리 리스크를 알 수 있게 되는 거죠. 실제적으로 이제 어세이 경험이 많거나 그러신 분들은 이 어세이에 대한 대충의 퍼포먼스를 예상할 수도 있고, 사전적으로 뭐 프라이어 놀리지나 아니면 우리의 경험을 통해서 이 하나하나의 액티비티의 그 리스크가 얼마 높고 낮은지를 알 수가 있습니다. 하지만 이것들이 이제 새로운 어세이고 경험이 없다고 하면 사실 모르는 거기 때문에 그때 리스크가 되게 하이가 되겠죠. 그 리스크에 비례해서 우리가 어세이 개발할 때 어떻게 이 부분을 테스트에 적용할 것인지, 사실 리스크가 높은 모든 걸 적용해야 되겠죠. 그런 걸 통해서 모든 조건들을 이제 QBD 컨셉에서 DOE를 적용하고, 이렇게 해서 어세이의 결과들이 어떻게 변하는지 확인해서 좋은 조건을 찾는 그런 방법으로 이제 어세이 개발을 하게 되는데요. 예를 들어서 지금 뭐 예를 들어 셀 덴시티를 조절한다. 이 부분은 이렇게 센터 포인트를 1 곱하기 10에 6승분 셀을 per ml로 집어넣겠다. 그럼 요 안에서의 뭐 50에서 150% 정도의 셀 레벨이 변했을 때 어세이가 어떻게 변하는지, 이런 것들을 확인해서 우리가 어세이 호난 어세이 개발에 어, 적합한 셀 퀀티티가 얼마 되는지 측정해 보겠다. 이런 것들이 이제 되겠죠. 이런 것들을 실질적으로 해보기 전까지는 리스크겠지만, 해서 결과가 나와서 어세이 퍼포먼스에 영향을 안 준다고 하면 리스크를 낮추고 관리에서 좀 벗어나서 어, 적용할 수 적용할 수 있게 됩니다. 그래서 지금 앞에 설명드린 각각 액티비티에 대해서 우리가 디파인 타겟 레인지가 어디고, 인베스티게이트 레인지가 어디고, 그걸 통해서 우리가 어떤 해석을 통해서 좋은 어세이를 만들 것인지를 대해서 결정을 하고, 결정을 하면서 개발하면 됩니다. 마찬가지로 지금 뭐 아까 말씀드린 도스 레스퍼스 커브에서 뭐 테트라졸린 솔트, 왜냐면 테트라졸린 솔트는 사실 이 어세이 같은 경우 카레메트리이기 때문에 이게 엔드포인트 어세이가 아닐 것이고, 그 다음에 이 테트라졸린 솔트 자체는 뭐 잘 아시겠지만 마이토콘드리아 디하이드로제이즈 액티비티를 측정하는 거기 때문에 거기서 시간별로 변할 거고요. 그 다음 그 양의 미컨트롤 하이드로에이즈 그 양에 따라서 또 다른 그, 그 어떤 색깔의 변화에 대한 그 키네틱이 있을 거기 때문에 어떤 양이 맞는지 뭐 이런 부분들을 우리가 세세히 확인하면서 세일을 개발해 주시면 됩니다. 자, 이 다음에는 이제 그런 액티비티들이 이제 완료가 됐으면 이제 어세이를 이제 어떻게 컨트롤 스트래티지를 가지고서 이제 진행을 할 것인가. 그 말씀드린 것처럼 모든 프로시저에서 우리가 위험하게 생각하거나 중요해야 되는 파라미터에 대한 테스트를 했을 거고요. 그걸 통해서 우리가 옵티멀한 조건을 찾았을 것이고, 그 다음에 사전 어세이에 대해서 그럼 우리가 이제 라이프 사이클 매니지먼트를 또 해야 되고, 그런 과정에서 당연히 데이터 트렌딩도 봐야 되고, 그러면서 보면 또 중요한 건 뭐 시스템 스테빌리티 해석하거나 아니면 샘플 스테빌리티 해석하거나 이런 일들을 하게 되죠. 그 모든 관용해서요. 저는 이 두 가지가 사실은 이제 그 트렌딩 할 때 가장 중요한 부분이라고 생각을 하는데, 시스템 스테빌리티 테스트에 있어서는 실제적으로 우리가 컨트롤하는 그 웰의 이 시그널을 보고 보면 이제 로어나 아니면 어퍼에서의 그 시그널을 봐서 그 시그널의 플라토가 어떻게 형성됐는지 이런 부분들은 이제 어세이가 잘 되나 안 되나 이런 부분을 잡는데 시스템 스테빌리티 활용을 하고 보통을 많이 하고요. 그다음에 실제적으로 이제 멀티펑셔널 그룹에서 막 리서치 쪽에서도 이런 일들이 종종 왕왕 있을 것 같은데요. 뭐냐면 샘플을 만드는 팀들은 당연히 자기 샘플들을 잘 만들었다고 생각할 것이고, 그에 따라서 이제 어세이 결과가 잘 안 나오는 경우에는 이제 그거에 대한 책임 소지를 따질 때 만든 팀과 또 어세이를 해준 팀과의 서로 이제 데이터 놓고 해석을 하잖아요. 이전에 한 번 나온 결과, 잘 나온 결과를 가지고 왜 그거와 다르냐 이런 얘기도 많이 하고. 그래서 뭐 GMP 에리어에서도 마찬가지겠지만 아니면 리서치 에리어에서 마찬가지고 우리가 이 어세이 받은 샘플들이 잘 만들어졌는지를 어세이의 결과에서 확인해 줄 수 있으면 그 어세이 그 샘플 만드는 쪽에서 좀 정확하게 얘기를 할 수 있겠죠. 그래서 그거를 위해서 이제 샘플이 잘 만들어졌는지 그 수터빌리티 어세스먼트를 하는 과정을 넣어주면 되게 좋은데요. 그때는 어떤 방법을 넣냐면 뭐 어퍼 어심톱 레이셔 어세이 그 고농도와 저농도에서의 그 레이셔 시그널 레이셔 기준을 예를 들어서 0.8과 1.1 사이 로어 어심톱 레이셔 다음에 또 어세이가 이제 이 익스포넨셜로 증가하는 그 부분에서 그 슬롭 그 다음에 또 어퍼 또 로우 어심톱 레이셔 뭐 이런 부분들을 적용해가지고 샘플 스테빌리티 어세스먼트를 하면서 우리가 어세이 퍼포먼스를 또 관리를 하고 또 이런 걸 통해서 또 어세이 결과에 대한 트렌딩을 하고 여기서 발견된 또 문제들을 다시 우리가 이벨리에이션 하고 이미 우리가 개발 과정 중에서 많은 것들을 해석했기 때문에 거기서 디파인된 리스크들은 결과들을 통해서 해석을 해서 리스크 미티게이션 하고 디파인되지 않은 또 리스크들은 새로 어세이에 추가를 해서 거기서 이제 그 부분들 프로시저를 다시 한번 어세이에 적용시켜서 더 옵티멀한 파라미터를 찾고 이런 과정을 계속 반복하면서 최종적으로 드럭을 잘 서포트할 수 있는 애널리티컬 메서드를 찾아 나가면 된다라는 거를 케이스 스터디로 말씀드리고 다음으로 케이스 스터디 그 두 번째 거에 대해서 김현준 님이 발표를 해드리도록 하겠습니다.
Okay. So this is a typical case study for bridging. And before we cover this topic, I think we need to understand why and when and how we do this. Obviously, when you're replacing something, then you have something that is insufficient, either requested by a regulator or something you know for sure that you have something better to replace with. We are handling drug product at this point, because we're at a stage where the program has been committed for drug development, going through a few phases of clinical trials. So there's a lot of investment, at least point of the company, there's a lot of investment there. So we have to ensure that how we do things and why we do things, and then when we do things, has to be justified, and it has to be justifiable to the regulator, where we could convince them that this is the right approach. So, and this is another example of potential use of historical data. I think Husang and Zurong covered or mentioned data trending or assay trending. Basically, the trending is used to evaluate historical performance, and then there are different places that you could use. And this is one of the areas that we could use it very well. So next slide, please. So the background is, as typical drug development, and once the molecule has been nominated as a drug candidate, I mean, company, get together and then they try to form a team where a lot of the analytical tool is being evaluated for development or support development. And I think it was mentioned by Zurong that, or even on a lot of the guidances, that you start with something that for sure that works. So in most times, immunoassays or binding assays, known to be more simple and more accessible, usually that is the first, I guess, approach to start with. But the reason is that bioassay requires some investment of time, because you have to understand the complexity of the MOA and how that MOA could, I guess, translate into your product quality when things do change during your manufacturing, as well as stability or storage conditions. So there are a little more investment involved in time in order to get bioassay ready. But I guess the best solution is to have them proceed at the same time, both of the technology, and then see what gets used for the attributes and the capability. In this particular case, I mean, nowadays there are a lot of technologies or assays or kits that you could use once you have a molecule. There are certain template of model or assay kits that you can use, but this work that we've done was done about 13 years ago, where no such kits or technology was available. So we had to generate and do all the research to make this happen. So the background is, in this particular program, we jumped into using the binding method to initially support potency or to demonstrate a drug quality. And as you know, the SPR is a sensor-based method where the binding or dissociation is being measured. For certain product, I guess that is okay, but depends on the your drug MOA or drug functions. You may need more than one activity to be monitored. So this particular case, since we are bringing up ADCC, it is a IgG molecule that requires functional expression of both Fab as well as FC portion. So where SPR is limited to just monitoring the changes or the format or modality of the Fab, whereas the ADCC could monitor both Fab as well as FC functionality. So that was one of the reasons, we need to do this exercise of bridging. And also at the same time, FDA definitely will get in and then start asking for, bioassay or more MOA reflective platform once you reach certain development or right around phase 2, phase 2B, that's when they will ask. So the limitation of the SPR is driving us to fulfill the role of potency testing with something more that will give you more reflection of the MOA. So ADCC method was being developed alongside for quite some time till this was actually requested by regulator for us to formally change. So the typical bridging study, obviously we need to have justification of why, and then we need to have definitely benefit of being better or equivalent. At the same time, the capability has to be demonstrated in terms of performance as well as capability of detecting product quality change. So next slide, please. So this is the, I guess the antibody. This is a anti-IL-5 receptor IgG1 antibody. This is actually a marketed product. I'm not sure whether you're familiar with Fasenra. It's an antibody that will bind to IL-5 receptor. This particular target cell is a eosinophil. And then once the antibody binds, it will help recruit the NK cells. And that would, once they're recruited together, the NK cells will elicit ADCC or cytotoxicity activity to the target cells, and it will deplete the target cells in your body. So as you can see, the SPR would only measure the fat binding portion. However, the FC binding is required to look at full structural function of the molecule. This is where it was actually being asked by a regulator to have the ADCC assay ready to switch. Next slide, please. And this is a, I guess, very simplified model of assay MOA. So let's start with the right portion of the diagram, where the CD16 activation on the NK cells will elicit few cascade of pathways. So as you know, the cytotoxicity is being driven by granzyme and porphyrin, but the effect of that granzyme and porphyrin will impact on the target cells, whereas the secondary cascade of activation of the effector cells actually is reflected from effector cells alone. So obviously we explored both areas. We did develop a method that reflects on the target cell killing, as well as the monitoring activation of the effector cells upon its binding with the target, with the drug intact. And I mean, this was more of a decision for, I guess, money and cost and time and ease. And I think Surong talked about heavily on the reproducibility, ease of use for QC, things like that. So those will be considered. So when you have multiple cascades or multiple pathways that you could establish, they're actually great tools to have both, because they're both useful for biological characterization for another different area. However, for the potency, you do want to pick the best technology that will give you least headache once it goes to, once it could support product lifecycle throughout commercialization. So in this particular case, we use the reporter. So the NFET cascade or NFET promoter with the reporter protein have been engineered in-house. So once again, at this time, we did not have any tools from Promega or anywhere else. So we had to do all this by ourselves because, I mean, there was nothing there at that time. Now things are much easier. So you could just buy a cell line from Promega and then do something similar. But I mean, this is somewhat different than using the surrogate model. This is the actual NK line being used to demonstrate ADCC instead of using a surrogate T cells. But anyways, on the left is a typical dose response curve of the reporter method that we had developed. So let's go to the next slide. I think we have a little more elaboration. So this is just an example of the method performance. This is a few aspects of method qualification where is written by ICH. You need to demonstrate linearity. So the left top, the diagram or graph is a linearity graph where you're graphing what has been assayed versus what is expected. So ideally, you would like to see a very straight line with a slope of 1. So, and this is used to demonstrate your capability of your assay, being able to determine different changes on the potency or changes of the drug strength. And then the strength is being changed linearly, not exponentially or other, I guess, kinetic. So you do want to see a linear kinetic for the change of the drug. And also at the same time, this actually defines your method range. So when you have a method, you definitely want to determine what are the qualified range of drugs that you're able to test. So this particular graph or this experiment could be supporting all those. One down, so left bottom, is the typical graph of a precision. So this particular method has, the assay format requires or use 3 plates. So each assay uses multiple plates where we have to demonstrate that there is no bias of the method or regardless of the sample position. So if the method is developed poorly, some, I guess, long assays or the assays with very long incubation definitely demonstrate some assay bias, we call it. But anyway, so we demonstrated that there's a high precision of reproducibility. The table on the top right is just an example of a accuracy. So you have simulated potency samples being tested at 50, 75, 100, and 125 and 150 strength, and we are assaying those values. So in any regards, the typical, in order to do bridging studies, you have to ensure that your method is capable of determining within the boundaries of the study. If you have a method that is very poorly developed, then these exercises will be pretty much a waste of time if the numbers that or data you're generating has very low confidence, where the study will become a little more complicated. But anyways, if you have done a very good job developing your method, retaining high precision, accuracy, linearity, and stability indicating property, then this package of the attributes could be used very well, doing more than just potency testing. So next slide, please. So this is a study plan. Mentioned earlier how the trending data or historical data is being used, and this is how we will fit that in here. So particular test or the bridging study has to demonstrate the performance attributes. So the attributes of the assay range, which has been defined through qualification that we could test it between 150 to 150 to 150 range. So those samples or simulated samples have been, the samples have been qualified for testing. So we know that each individual samples are not contaminated at each experiment. But anyways, that will generate a precision of performance. At the same time, we use a actually released lots that's been released in the past. So that will also generate lot-to-lot variability, as well as a test to demonstrate stability in the indicating property. So this is one of the key attributes of the potency assay. But as you can see here, this is actually very minimum design. We've done multiple bridging studies in the past. At least we have done this work not too late phase, where we weren't required to do a lot more. For this particular, I guess, example or particular case, or the bridging study as a exercise, the later you do it, more work is involved, as well as more data needs to be generated. So just keep that in mind. So earlier you have things ready better. And hopefully when FDA asks to have these activities being pursued, your lab should be ready to perform these tasks. So if you do not have a method at the point they're asking, then you're talking about year to 2-year delay in the program, because you won't be able to demonstrate these, because FDA will not take binding assay as a release. So you have to I guess counter risks from this point. So next slide, please. So this is the design, the parameters that we picked. So the particular bridging study, you preset the parameters based on the method capability. You do not calculate the numbers after the testing is done. You actually commit into the parameters and precision before the study begins. In order to be successful, you need to use your historical data to evaluate. So this is where the exercise of using the historical data, simulating the study to make sure that your normal performance of the method could meet such criteria, because the failing actually will be very detrimental. This is where you want to make sure that you know your assays, you know how it I guess behaves in terms of in the actual laboratory setting. And at the same time, these analysis, even though a lot of times among scientists, we all think that we could do at least some basic level of statistics. So you could use your Excel spreadsheets, you get your standard deviations or whatever confidence interval that's calculated by Excel. But since this document will be submitted to regulator, I think it would be much easier to consult with biostatisticians and then get their certificate or their report. So this is where we actually work closely with Harry Yang, when he was leading a group or stat group at AZ. So we work very closely with them. We have to educate them, or you have to educate the statisticians what the limits are, so they could properly give you the scenario of exercise for simulation. But anyways, so please work with statisticians instead of trying to get this do it through Excel, because once you write a formal report, there will be less to explain. If you get the professionals, even though we are professionals, but professional in the field to do it. So this is one other way to relieve some of the potential headaches. So the actual limit is pretty simple, because we're looking at the equivalence limits of 25% with a 95% confidence interval. And we also want to design the study where the study or the experiments are challenging the method for 80% success rate to show the difference that is bigger than 10% difference. So, these are typical exercise for doing such study. And then also at the same time, the capability of the method, and then we focus on the stability indicating property. And the sample size calculation, once again, this is based on the power analysis of your historical data. So once again, you need to justify where these numbers are coming from. So if you have these data, or if you have these historical data, then use them. It will be much easier to justify your study. Otherwise, then you will be explaining a lot of things, because you don't want to just come out with a number without any justification, especially because you have to present this to regulator at the stage. So next slide, please. So this was the output of the exercise. You can see here the method range, the testing for the 50 to 150%. We hit, within the 90% confidence, we're crossing zero, meaning that the method is equivalent. At the same time, the bar is not going outside of the 25 confidence interval, or 25% estimate. And the lots that we tested at the same time, we're getting similar results, is crossing zero, and then we're not seeing limits of 25%. So this particular set of data demonstrates that both methods are equivalent or similar. So we're good here. So next slide, please. So now this is something that only Bioassay could detect. As we mentioned earlier, this particular molecule has a bispecific function. So you have the FAT function that binds to IL-5 receptor, as well as there's the FC function where that requires binding to CD16. So in the case where one of the active area have been impacted by some form of process change or stability or whatnot, then it has to be, your method needs to be sensitive enough to detect that. But in our case, the SPR was not able to see any difference in this case of the sample or the sample degradation. So what we're assuming is that the degradation is actually happening at the FC glycan level, where there are changes. So, it's a simple graph, so you could tell there are differences, but in terms of the product quality as well as patient safety, this is actually pretty bad data, or I wouldn't say it's bad data, it's bad case, where when you have so in the case of this particular degradation, it seems to be that FC portion is being impacted by, I guess, the room temperature. Obviously, it's very extreme at three years. So if you're familiar with enzyme kinetics, you probably have better understanding what happened, what is really going on when your upper asymptote or maximum changes. So what this really means is your degraded drug is actually inhibiting your non-degraded drug. So that's why you're seeing a maximum changing. So this actually is bad. It's much better to have a less active drug than inhibiting drug variant. So this becomes a inhibiting drug variant. But anyway, so at least we could demonstrate that the bioassay seem to be much more sensitive to product quality change. So once again, the study goal is to demonstrate equivalence in performance, so which means your precision assay range has similar output or similar expected performance. But then at the same time, the capability of the method of detecting, or sensitivity of the method for detecting the drug functional change. And then that this is actually what the difference is. So bioassay is sensitive to drug quality change, whereas the binding assay was not. So next slide, please. So, this was basically what we wrote off as justification. So after, the study was performed, we're justifying that the ADCC or bioassay in this case is I mean, even for hypothetical, theoretically more MOA reflective, because it requires or is monitoring both the Fab as well as FC end of function. And ADCC reporter-gen bioassay has demonstrated equivalence in performance in this particular study, both using assay range as well as the different lots that we have tested. And on top of that, the method or bioassay seems to be more capable in terms of monitoring stability indicating property. So we were able to replace the method from SPR. And we did actually submit this packet to FDA. And we did not get a single question from them. So everything went very smoothly. So next slide, please. So in summary, as you can see, the bioassays are very challenging because, you know, it has very complex product-specific mechanism. And a lot of times, especially when you're starting with the program, even within the company, there aren't as much information about the molecule or the MOA or the pathways. So, I mean, that is one of the biggest challenges for starting to develop bioassays. And as for the point of regulators, I mean, they're required. I mean, I'm thinking that there should be more aid or help, especially for the companies that aren't very experienced. And at the same time, there has to be a bar of standards that people should be able to look at as an example. You can see a lot of the regulatory documents or some of the guidances. They seem to be very generally, I mean, it's very general framework, but it doesn't give you much information other than, you know, you hire a consultant like us, I guess, because, I mean, us being involved with Amgen as well as AZ, when we joined AZ, actually, the knowledge of AZ for this particular area wasn't that advanced or experienced. So even, I mean, so there were challenges trying to establish certain function for supporting these type of drug development activities. But anyways, even when you look at one of the newest guidance, I think the Q14, I think came out either this year or last year. I mean, it's not, I mean, there's very good, it covers big areas of development, but it doesn't give you specifics in terms of example of, you know, how you should do things. But anyway, I think this is the most important part, is that, you know, to ensure, you know, quality of biopharmaceutics, because this is not, I mean, we're talking about, I mean, it depends on how you, or when you look at it, we're talking about protein, but this is a protein that's actually being used by people. So, I mean, we call it a product, but this actually product is very essential, where if you miss a product change, I mean, it could end up in very high risk situation. So this is where, you know, you want to do as diligent work to get the most or have the most understanding of the molecule as possible. And having or having the technology ready for the company as early as possible is very critical. Once again, for a lot of smaller companies, you know, when these things are being requested by regulators, it's too late because they are not ready, because they rely on, let's say, immunoassay to start off, and then, you know, when the regulator steps in and asks, and you do need time to be ready. And then the time to ready is pretty long if you're, especially when you're working on something that no one else is doing. So when you have innovative molecule, is more important that this work gets started early as possible. So thank you. That's all of my talk. So I would pass it on to Husan for continuation.
네. 아, 이것으로 저희 발표 슬라이드 마치고요. 발표 중에 제가 듣다 보니까 하나가 더 전달해주고 싶은 이야기가 있어서 잠깐 설명드리도록 하겠습니다. 저는, 어, 이전에 이제, 제가 분석 관련 전체를 할 때가 많았었는데, 그럴 때 보면 이제 저희가 어세이 한 60개 정도 프로덕트 서포트 할 때 개발을 합니다. 또, 캐릭터라이제이션 할 때 사용도 하고. 그중에서 가장 항상 걱정이 많이 되고, 결과들을 보면서도 항상 신중해 봤던 게 이제 바이오세이, 포텐셜 세이, 뭐 바인딩 어세이 이런 것들이죠. 대부분 다른 것들은 베리에이션이 적고 또 결과도 굉장히 프라미싱하게 나오고 또 굉장히 약물 자체가 나타내는 결과들이 굉장히 내로한 레인저에서 움직이니까 그런 점이 있지만 요건 좀 그렇지 않고 좀 흔들린 경향이 많이 있고 근데 제가 하나 더 말씀드리고 싶은 건 뭐였냐면 이제 이 그래프는 지금 Stability Indicating Property 뭐 이런 부분을 설명하려고 설명을 했었는데 실제로 이제 어, 여러분들이 결과를 어세이를 개발해서 테스트하다 보면 이런 비슷한 결과들을 보실 때가 종종 있을 거예요. 근데 지금 이거는 스트레스 샘플이 이래서 그렇지 만약에 이게 레퍼런스고 이게 테스트 물질이면 이 결과를 어떻게 해석을 해야 될까? 아마 이거를 경험이 없으신 분들은 이 두 개를 컨스트레인 모델을 만들어서 인위적으로 이걸 같은 물질로 여기고 거기 안에서 상대적으로 렐러티브 포텐시를 구하는 방법을 갈 거거든요. 그렇게 되면은 이 물질이 서로 다르다는 걸 확인할 수 없겠죠. 근데 이 정도의 차이를 그럼 과연 어떤 지금 RLU 유닛의 차이를 과연 다른 물질로 볼 것이냐, 같은 물질로 볼 것이냐 그런 부분을 해석해서 다를 때, 안 다를 때 이거를 가지고 우리 물질이 퀄리티가 바이어스 됐다, 안 됐다 또 이런 것도 해석을 해야 되니까 네, 단순히 아까 말씀드린 것처럼 이거를 인위적으로 컨스트레인 모델을 만들어서 포텐시 밸류를 해석하고 그 밸류를 가지고 리포팅을 하고 이렇게 되면 나중에 뒤쪽에 갔을 때 지금까지 개발했던 결과들을 다 믿을 수 없는 상황이 될 수 있는 가능성이 높기 때문에 이런 어세이를 실제적으로 하시는 분도 그렇고 또 이 에세이를 보고 리뷰하시는 분도 그렇고 또 정리해서 리포팅하는 분들도 그렇고 이런 내용들을 잘 살피면서 데이터를 이밸류에이션하고 또 그거를 트렌딩하고 해나가셨으면 좋을 것 같습니다. 어, 저희는 지금 보이는 슬라이드는 저희가 이제 아스트라제네카 또 암젠에서 개발된 약물들, 뭐 파스나, 뭐 많이 들어보신 거는 시너지스, 어, 임주도, 뭐 이런 분들 많이 들어보셨을 것 같고 이런 약들. 암전에서는 블링 사이트, T셀 인게이저, 그다음에 더 많이 아시는 엠버, 그다음에 요즘에 뭐 셀트리온도 잘 만들고 있는 프롤리아 이런 애들, 그다음에 바이오시밀러는 아바스틴, 릴툭산 이런 부분들에서 저희가 이미 허가를 받은 16개의 어, 제품 허가를 저희가 서포트를 했고요. 플러스 이거 말고도 개발 중이거나 아니면 중간에 이제 드랍된 약물들이 있으니까 훨씬 더 많은 제품들을 서포트를 했었습니다. 좀 과정에서 네이처 드럭 디스커버리의 FC 팩터 펑션 어세스먼트 관련한 거, 그거의 컨트롤 스트레리지, ADC 리포터 프로세스 저스의 테라포트 안티바디, 그다음에 또 GMP 컴플라이언트한 오토메이트한 포텐시 어세이 템플레이트 이렇게 하고 그걸 어떻게 분석할 건지 그런 부분도 저희가 PDA에 됐고요. 그리고 최근에는 아직 퍼블리시는 안 했지만 카티 LVB 벡터 이런 거 활용함에 있어서 어떻게 지금 쓰고 있는 방법이 어떤 가지고 있는 한계점 그걸 어떻게 극복할 것인가에 대해서 어, 새로운 대안을 제시하고 결과를 또 내고 했던 그런 부분을 제가 뭐 조만간은지는 모르겠지만 퍼블리시할 계획이 있습니다. 그래서 저희가 이런 경험을 바탕으로 오늘 말씀을 드린 거니까요. 이런 얘기를 잘 어, 어, 이해하시고 앞으로 의약품 개발에 적용하시면 좋을 것 같다는 거를 알려드리고요. 그다음에 혹시 이 일을 하다가 이제 어, 이 부분도 되게 전문성이 많이 필요한 부분입니다. 저희가 어, 셀진 테라피 관련된 캐릭터라이제이션, 타이틀 퀀테이션, 관련된 DSTP 릴리즈 메서드, 당연히 뭐 안티바디나 이런 것도 관련된 것들은 뭐 포텐셜 릴리즈 메서드, 스테빌리티 테스트 등등, 또 바이오시밀러 프로그램, 또 도시어 이런 부분도 저희가 많은 경험을 갖고 있고 서포트를 할 수 있고요. 그다음에 CMC 컨설팅 관련해서는 아까 말씀드린 것처럼 퀄리티 매니지먼트 부분들을 또뭐 같이 해서 그래서 이런 분들 저희가 저희 솔루션을 갖고 있는 부분이라서 혹시 나중에 저희 도움이 필요하시면 다음 주소로 연락 주시면 제가 최대한 도와드리도록 하겠습니다. 네,
네,
이상으로 발표를 마치고 질문이 있으시면 받도록 하겠습니다.
네, 발표 잘 들었습니다. Thank you for your insightful presentation, Dr. Jiang and Dr. Kim and also Dr. Huang. 이게 가장 이제 핵심적인 질문이 될 것 같은데요. 먼저 지금 이제 말씀 주신 그런 케이스 스터디들이 실제로 미국 FDA에 제출되는 자료로 활용 가능한 정도의 결과인지 궁금합니다. Other case studies of FDA submission quality가 질문이 되겠습니다. 첫 번째로요.
네, 아까 저희가 보여드린 것처럼 이미 저희 16개 이상의 약물들의 허가를 받았고요. 그때 이제 저희가 서포트를 했었고 지금 어 지금 어세이 개발부터 그 다음에 지금 나오는 그 라이프 사이클 매니지먼트 이 부분들이 다 말씀해 주신 것처럼 이제 허가 자료에 반영이 돼야 되는 부분이겠죠. 지금 말씀드린 그 부분은 그거를 모두 커버할 수 있는 부분에서 말씀을 드린 거고요. 네.
네. 그 의약품의 제조 프로세스 중에서 어느 정도까지 커버리지가 이제 가능한지가 궁금합니다. How much coverage does your service provide in the drug development processes?
Zorong, you there? Or you want me to cover?
Yeah,
So,
coverage.
Sure,
for-
Jung, you
Okay.
go ahead.
Okay, so basically, I think even in the US, the strategy has been changing. But just to give you a simple, short answer is normally our area of experience is from right after the drug discovery. So in most companies, we call drug candidate. That's before tox. So
Before tox.
before tox. So once the molecule has been identified as a drug candidate, so we call it candidate drug or CD. That's when you start communicating with the CMC team, I guess at that point, because you want to look at developability in terms of the drug. So our involvement normally is from that point all the way up to commercial. So which is pretty wide. So so we could support up tox, which means we get involved before tox to provide platform because when you do like formulation, I would say, or heat study, or because a lot of the initial work with the formulation group, because they change formulation and you want to understand what these chemicals are doing to the molecule. So they do formulation alongside with the, I guess, the heat storage. And then sometimes different chemical composition has vastly different impact on the molecule. So yeah, we could start usually from there, which means that, I mean, we have a platform already that could support most of the product, unless you have some really complicated chemistry, which may require time, which means that it's better to start earlier, right? Because you want to be ready. But anyways, yeah, we could start from early to the commercial.
Yeah,
Thank you.
And
Just at this point,
Yep.
so the early development largely depend on the product mechanism of action. So, let's say a class 3 molecule where mechanism of action simply involve a monoclonal neutralize a soluble ligand. Most companies, they probably could use those affinity-based, those binding assay, where probably you could select the right candidate. But in the case where your product mechanism of action involve FC effector function, or some company have a specific strength to do some FC engineering, where different kind of a glycol form that could impact FC effector function. So in this case, you might experience a pure binding assay might not fully reflect product mechanism of action. And if you do those kind of a screening, select writing clone at this stage, you might miss the one where they have a better efficacy profile. So, so it's really depend. Like, so some of the molecule like T cell engager, some other one, you might have, if you have a function assay, you might not miss some of those like a receptor binding, occupancy, like internalization, or any of the transformational change where binding might not be able to detect. So we definitely have a toolbox for different state, different variety of modality molecule at development phases. We could at least provide some feedback through consulting, because sometimes those clone selection might be critical when you have the right assay in place. Yeah.
So, I mean, just to clarify, I mean, we're showing examples of a bioassay, but in, I guess, the US, we call this area of potency. And then usually potency assay covers, I mean, bioassay to be more effective because it's more welcomed by regulator. But we also develop a lot of immunoassays as well. So, I mean, I guess, just to give you an example, if you want to look at bispecific proximity, then you could, I mean, we have a lot of experience using alpha screen as well as, a TNF, no, not TNF, what is that? The
TRF.
Yeah.
TRF.
TRF. So in our area, our expertise is more, I mean, we classify as a potency. So anything that is required for potency, whether using cells or chemicals, I mean, that's our expertise.
Yeah. 제가 이렇게 발표를 들어보니까 바이오어세이, 기본적인 바이오어세이의 중요성이 굉장히 우리가 간과하는 것 같은 생각이 듭니다. People talk a lot about the importance of toxicity or clinical trials, but I guess the importance of the basic bioassay is sometimes underestimated, I think. And I think modalities are becoming more diverse from antibody, just only antibody, to ADC or CAR T, cell-based therapies, and something exosome something. So do you have any solution for the new kind of modality drugs?
So I could chip in, then June could add on some of those. So the challenge there in the industry is a different level. Firstly, like the reference basically is important material used to understand your clinical material or for your PPQ beyond. You always have to establish a reference standard. And this is pretty well established in the therapeutic monoclonal area, ADC area also, with exception of the ADC, you got a naked antibody with a linker, with a payload, where you're trying to dissect and understand, but you still need a reference standard with the same principle as a therapeutic monoclonal. But in the cell gene therapy space, relatively speaking, the reference standard is getting much more challenging. starting with AAV virus, where you have a different kind of working group, different serotype, like AAV serotypes 6, 5, and 9. Depend on the subtype. So some of those they don't have are is type of reference standard. If you move to the CGT era of CAR T, so in general, CAR T does not have a good references because autologous CAR T, you mind dealing with lentivirus where you could have a small batch characterized as a reference. But again, a lot of those assays used there, like a titer assay, very variable. So not even stability indicating. So even the CBER, like Dr. Peter Marks often say that there's a lot of company, they have a hiccup because lack of a reliable potency there. So we do have a solution where produce a functional assay that's stability indicating, where we could monitor the activity precisely for the lentivirus vector, which in the CAR T space is called a drug substance. But when moving to the drugs product for the CAR T, that's another challenge because each individual patient itself a product. So you cannot just generate a universal reference for all different patients. So that's kind of a whole challenge in the whole field. Maybe for the allograft, if it's engineered allo CAR T cell, universal CAR T. But again, universal CAR T has their own challenges because their efficacy profile not comparable to the autologous. Most approved product currently by FDA, they all autologous CAR T. So it's really depend on modality molecule. But
Yeah.
I think we have the experience to handle all of them.
Yeah, thank you for the answer. And 혹시 궁금하신 점이나 질문 있으시면 남겨주시면 됩니다. My last question is the AlphaFold 3 from the Google has been released just only 12 hours ago. So what do you have any idea of the challenge of bioassay, basic bioassay in the era of the in silico?
So, the drug discovery is evolving pretty fast, including those AI-assisted drug discovery. So some of those technology got more proven in the discovery stage, whereas the others in the application drug release, it yet to be realized. Probably I will say a couple of years behind. So I'm as one of the organizing committee members still with the CAS, where we have a lot of FDA, European agency, Health Canada agency on the organizing committee. We still organize the Bioassay conference each year in US. As far as I'm aware, virtually most bigger companies, the larger company like Roche, Genentech, Merck, Eli Lilly, all the Amgen, AstraZeneca, they all attended those conferences. I have not seen anyone yet using those kind of in silico approach.
Yeah.
Most of those in silico approach really they use for those immunogenicity predicting, those hotspot immunogenicity assessment. I know some CDMO like Lonza, some other, they do have those kind of program where it's a fee for service, depending on the number of molecule, you could do that. But I'm not aware in the product release, this has yet to be realized. So,
well, I think this is similar to using like biostatisticians. So you guys know for sure, or many companies' culture is to work closely with the biostatisticians. But then if you go to the conferences, a lot of times people bring out their data saying, oh yeah, we work with biostatistician and we perform the DOE for certain
or bioassay development or such. The key issue is, you give it to biostatisticians, which they're good at juggling numbers, but they do not understand the science of what is critical and what is not. So they end up generating a lot of data, and then they do come up with a range. They do come up with the proposal for what is best, but in the eye of someone who's experienced, a lot of times they're knocking on the wrong door, kind of, because the setting these exercise of experiments only useful when you understand the critical parameters, and then what the range is, or understanding the useful range is usually by experience, not by just crunching numbers. So I mean, definitely the AI will probably get smarter, and who knows whether they have eye for these things in the future. But at least for now, you seem to be, I mean, I think it will take time. So, I mean, this is also similar to, let's say, automation. And then let's say if you have a method, and then you get automation engineers to program your assay, usually it doesn't work. I mean, many companies have tried that, but it failed. So at least from my experience, having scientists to learn automation is successful, not having engineers to learn bioassays.
Yeah, that kind of results of the in silico model should be in the track of the drug development. So maybe our potential customers should be increased, I think. Maybe. I hope so, actually. Yep. There is no question for now, so we're gonna end up so far. So, do you have any comments for the finishing the stage? No?
네. 아까 김학준 대표님 말씀해 주신 것처럼 셀 베이스 어세이가 아니면 바이오 어세이가 사실 학교에서 공부할 때나 이때부터 계속 많이 사용하는 툴이잖아요. 그래서 되게 쉽게 지나칠 수도 있고 누구나 잘한다고 생각할 수도 있고 한데 이거를 인더스트리에 가서 약물 디벨롭먼트 서포트하기 위해서는 또 다른 해석이 필요하고 또 다른 전문성이 필요하다고 생각이 듭니다. 그래서 그런 부분들을 잘 고민하면서 약물 개발에 적용시켰으면 좋을 것 같습니다.
네, 감사합니다. 지금까지 발표해 주신 황우상 박사님과 닥터 지양, 닥터 킴께 감사드립니다. 네, 이것으로
네, 고생하셨습니다.
이번 세션을 마치도록 하겠습니다. 감사합니다. Thank you.
Great. Thank you.
Thank you, June, your time.
Bye bye.
Yeah, you have a wonderful rest of the day. Thank you.
Thank you.